Hello, boys and girls, and welcome to Mendelian Genetics. This PowerPoint is going to go over Gregor Mendel, the monohybrid crosses, and Mendelian genetics, which means one trait is coded for by one set of alleles, by one genotype. So the traits that we're talking about is one gene codes one trait, and that's it. So on this next couple of slides, you'll see a couple of vocabulary terms. For example, right here you see genetics, which is the scientific study of heredity, character, a heritable feature, a trait, the variant of that character, true breeding, plants that self-pollinate, and all offspring are the same variety, allele, an alternate version of a gene. Okay, this one's really important because you can have one version of the gene or another, and that's the allele. Do you have the A version or do you have the B version? Do you have brown eyes or blue eyes? Do you have hitchhiker's thumb or do you have a straight thumb? And that's an allele. The allele is the code for those traits. Dominant allele, an allele that is expressed and it masks the other in a heterozygote or a homozygote. This means that this is the trait that is going to show. If heterozygous, this is the trait that shows. A recessive allele, this is an allele that is present but remains unexpressed, or it is masked in the heterozygote. Homozygote, this is a pair of identical alleles for a character. If it is homozygous dominant, it would be a capital B, capital B. If it is homozygous recessive, it is little b, little b. If it is heterozygous, then it's going to be two different alleles. It'll be a big B or a little b, and the dominant trait is going to show if it is in Mendelian form. Genotype, this is the genetic makeup. It's the combination of alleles that an organism has. And a phenotype is the appearance, the physical traits, the characteristics are determined and by the genotype with the environmental influences. A monohybrid cross, this is a cross that tracks a single character. P generation, the parent or the true breeding. F1, first filial. These are the offspring of the P generation. F2, second filial. These are the offspring of the F1 or the grandchildren of the P. Gregor Mendel was an Austrian monk. He was from Austria during the 1800s, the 19th century. And he studied pea plants, looking at these pea plants, and which was really smart of him to do because pea plants have these great things about them as one, they take a really short time to germinate. Two, they are self-fertilizing, meaning that they are going to fertilize themselves, and every children that come from them come from that parent pea plant. So all the traits are going to be exactly like the parent before it. And they have these characteristics that are really easily observable. And using what these traits and what he saw, he developed these principles, and this is why he's called the father of genetics. And the traits that he studied were these 7 traits, which was pea color, and they were either green or yellow, pod color, which was green or yellow, pea shape, round or wrinkled, pod shape, whether it was inflated or wrinkled, flower color, which was purple or white, and plant height, which was tall or short. You see that there's those 4 on this slide, but remember that pod color and pod shape were also part of them. And he made these observations with the example to the right being the pea shape. And when he crossed a round pea and a wrinkled pea, he saw that he always got round peas in the F1 generation. And when he allowed those F1 generations to breed by themselves to self-fertilize, he saw that he did not get the all round peas. He got 3 round peas to 1 wrinkled pea. So he got that 3-to-1 ratio. Now, how he made sure that he got the crosses that he wanted and did not get crosses that he didn't want was he would remove the stamen or the male parts of the plants. So he would remove the stamen from the purple flowers, he would remove the stamen from the white flowers, and then he would transfer the pollen from the white flowers to the purple flowers, and vice versa. No, however he did it. And he would look and watch as these plants would germinate, grow seeds, and then how they would create new life. And so for the purple ones and the white ones, he saw that by transferring the pollen from the white flower to the purple flower, he got these matured pods and these planted seeds, and he got all purple flowers. So the purple flowers, instead of getting, at the time they thought it was a blending principle, which means that you would get a middle characteristic. So he was looking and he thought he was going to get light purple flowers, but that wasn't how he got it. He got all purple flowers exactly as purple as its parent. Now, these pea plants have several advantages for genetics. They are available in many varieties with many distinct heritable features with different varieties. And the advantage is that he has very strict control over which plants mated with which because he removed the male parts of all the flowers. And that in nature they self-fertilize, which means that no matter what happens with those traits, he's going to be able to see those traits over and over and over again if they're pure breeds. And if they're not pure breeds, that's when you're going to get that variety showing up. And so because he could remove the stamen, he could cross-pollinate plants. So in a typical breeding experiment, Mendel would cross-pollinate or hybridize 2 contrasting true breeding pea varieties. So he would get the parents, which are the pea generation, and those are our pure breeds. And then he would crossbreed them and get the F1 or filial 1 generation, which would be hybrids. And then he allowed those hybrids to self-pollinate and get the F2 generation. And due to the quantitative analysis of those F2 generations, we got 2 laws, the law of segregation and the law of independent assortment. Now, the law of segregation states that when gametes, so sperm and ovum, are formed, each gamete will receive one allele or the other. Now, this is really important because in meiosis, this is how we separate the chromosomes during metaphase 1. Those chromosomes line up on that metaphase plate, and then we separate the chromosomes in anaphase 1. So between metaphase 1 and anaphase 1, the law of segregation is what says how we split them up. And the law of independent assortment says that 2 or more alleles will separate independently of each other when gametes are formed. So the alleles are going to separate independently. They're not going to, like, the alleles are not going to move with each other or away from each other. They are going to be independent of each other because they are on different chromosomes. So the law of segregation works with the law of independent assortment on metaphase 1 and anaphase 1 because the chromosomes are lining up and they're separating independently. So the law of segregation states that the 2 alleles for a character are packaged into separate gametes. And if the blending model was correct, we would get light purple flowers from the purple flowers and the white flowers. Or if the blending model was right, when we bred a tall plant with a short plant, we should get a medium-sized plant. But that's not what happened. Instead, we got all tall plants because tall showed up more often than short. Or purple flowers showed up more often than white flowers. Now here we have a Punnett square, and genetics problems can be solved easily using this tool. This was made in the 1900s by a man named Punnett, and this is a great tool for calculating genetic probabilities. And we use this, I'm going to show you real quick, by calculating these tools. Now I'm going to use the letter T, and this is going to be for tall. And tall is dominant over short. Now, we're going to do a parent generation. So a P generation, this means that they are homozygous dominant and homozygous recessive. Remember, homozygous, homo means same. And when we cross them, the columns are going to go down and the rows are going to go across. So here we got 4 out of 4, or 100% tall children. So this is a 1 to 0 ratio. And this gives us all the information of this gives us a lot of information of what we need to know here. All the children are tall. Now, using a several step process, we are going to look at tallness is dominant over shortness, which is kind of what we just did there. A homozygous tall plant is crossed with a short plant, and we're going to look at the offspring. The phenotype, what is the phenotype? Remember, phenotype is similar to physical, pH for phenotype, pH for physical, and genotype, geno for gene, gene your DNA, and we're going to use alleles to write down how these are, like what they're going to be. So here we have the alleles for tall plants on the top and the alleles for short plants on the side, and we want to make sure that they are all on one side together. So if we look at this, one parent is going to be on the top and one parent is going to be on the side. We don't want to make them across from each other. They want to be on the same side because this tells us which plant is related to which. So we're going to take each allele and separate them along the top or the side of the Punnett square. Now, we're going to write the alleles across each column or down each row, and the resulting mix is the genotype of the offspring. So here we have all heterozygous, and they're all going to be tall. So they are heterozygous for our genotype. So our genotypic ratio is going to be big T, little t, 100%, and they're going to be all tall for the phenotype. So if you're asked a question, what is the genotype? You're going to say big T, little t. And if you're asked a question for the phenotype, you're going to say all tall. So we're going to take this a step further and we're going to look at it through the F1 generation and their possible kids. Now, if we look at this first top left box, you're going to see that it is a homozygous dominant, so big T, And if we look over on this bottom right hand box, we see it is homozygous recessive, or little t, little t. And if we look at the 2 in the middle, we see that it is heterozygous, big T, little t. Now, from there, when we look at it, we see that we have 3 that are going to show the tall trait, and 1 that's going to show the small trait. So we have a 3-to-1 phenotypic ratio, or a 1-to-2-to-1 genotypic ratio. Now, if you look, we get the 1 red circle, 2 in the orange circle, 1 in the purple circle, so that's our 1-to-2-to-1, 1 big T, 2 big T, and 1 little t. And then our 3-to-1 is 3 tall to 1 short. So that's some of the interesting things. We have a 3 different in our genotypic ratio, 1-to-2-to-1. And if you add them all up, it equals 4. And then we have 3-to-1 in our phenotypic ratio. 3 plus 1 is 4. So 4 things in our boxes in the entire thing. So we can look at it in a ratio that way. So when Mendel allowed the F1 plants to self-fertilize, he saw that he got a 3-to-1 phenotypic ratio. And he was able to look at this by his large sample size and by the quantitative analysis. Remember, quantitative, it sounds like quantity, which is it has to do with numbers. And he recorded 705 purple flowers to 224 white flowers, and that essentially gives us that 3-to-1 ratio. And that white trait that disappeared in the F1 children showed up again. So it's not that it was gone, it was hidden, it was masked. So the purple trait had to be dominant so it could mask the white trait, which had to be recessive to hide. Which here we have our other picture. You see right here we have the purple and white flowers over here in the parent generation. You have our F1 generation where all of them were purple. And then you see that in our F2 generation, we have our 705 purple flowers to our 224 white flowers, giving us that 3-to-1 ratio. Now, the law of independent assortment says that each set of alleles will separate independently of each other. So if we have a 2 to the n, we have 2 to the n from the law of segregation. So 2 to the n possibilities. So here we have all of these things where it says each set of alleles separate independently of each other. So that we have big G, little g, and they're going to separate independently from big Y, little y, little y. So if we look at this first section right here, you see that we can get big G, big Y, little g, little y, little g, little y. We can get a big G, big Y, or big G, little y, or a little g, big Y, little g, big Y. Now, a test cross is designed to reveal the genotype of an organism. So we use a known genotype, usually the recessive, because it has to be little p, or little g, depending on whatever it is. And then we use the unknown genotype, which is usually our dominant, and it's usually we're trying to see whether it's homozygous or heterozygous. Now, if this is homozygous, then all of the offspring have to be purple. Because no matter what happens, a homozygous purple plant and a homozygous white plant is going to show with the all purple trait because they're all going to be heterozygous. Where on this one, if we have one heterozygous and a homozygous recessive, when we do the cross, we get two heterozygous, which are going to give us that purple plant, and two homozygous recessive, which is going to give us that white plant. On our next lecture, we are going to have a dihybrid cross where we are going to cross not one trait, but two traits. So make sure you come and tune in and you watch this dihybrid cross because for some people it can be really tricky and we're going to try to break it down in a way that is going to be simple and easy for you guys. See you next time.